Welcome to AP Biology. This video is an overview of cell respiration. Cell respiration is an exergonic catabolic pathway. To be an exergonic process means that it's energy releasing. During cell respiration, organic molecules found in food, are going to be broken down in order to regenerate ATP. And that's our major goal here, the regeneration of ATP. We typically think of glucose as entering cell respiration, but many different carbohydrates can enter, such as fructose and others. Amino acids can enter cell respiration. Fatty acids could also enter cell respiration. On the bottom of the page, we see the typical equation for cell respiration. We have glucose plus oxygen are going to yield carbon dioxide, water, and energy. Since energy is released, this is why we call cell respiration exergonic or catabolic. Cell respiration is going to involve a series of redox reactions. Redox reactions involve the transfer of electrons. One molecule is going to get oxidized, which means it loses its electrons, and the other molecule is going to get reduced, meaning it gains the electrons. During cell respiration, overall, glucose is going to get oxidized. It's going to have its electrons taken away. When glucose gets oxidized, it becomes carbon dioxide. When glucose gets oxidized, its electrons are going to be transferred over to oxygen. Oxygen gets reduced and it forms water. Remember that whenever a molecule gains electrons, it's also going to gain protons with it, or it's going to gain hydrogen ions. So when oxygen gets reduced, oxygen is going to turn into H2O water. Oxygen, overall, is the major oxidizing agent of cell respiration. It's pulling the electrons away from glucose. The reason why oxygen pulls electrons away from glucose is because of its high electronegativity. Remember, electronegativity is the pull of electrons. The first process in cell respiration is always glycolysis. Glycolysis happens no matter what, whether oxygen is present, whether oxygen is not present. During glycolysis, glucose is going to enter the cell, and in the cytosol, glycolysis is going to take place. Glucose is going to be cut in half to form 2-pyruvate. During glycolysis, overall, 2 ATP are going to be made, and more importantly, 2 NAD+ are converted into 2 NADH. NADH is an electron carrier. When glucose gets oxidized during glycolysis, its electrons are removed. Those electrons get sent over to NAD+, so NAD+ gets reduced and it forms NADH. NADH is going to function in cell respiration as an electron carrier. It's literally an electron shuttle, and we'll see in a little bit that NADH shuttles all the electrons that came from glucose to the electron transport chain. This whole process of glycolysis is happening in the cytosol outside of the mitochondria. In this diagram, we see what the mitochondria looks like. Here's the outer membrane of the mitochondria. Here's the inner membrane of the mitochondria. In between the two, we have the intermembrane space. And in the center of the mitochondria, we have the matrix. So we have two scenarios we're going to play out. What can happen if oxygen is not present, and what can happen if oxygen is present. If oxygen is not present, aerobic respiration will not take place. Instead, a separate process called fermentation is going to happen. If oxygen is not present, pyruvate does not enter the mitochondria. Instead, it stays in the cytosol and it enters fermentation. In human muscle cells, lactic acid fermentation is going to take place. During lactic acid fermentation, pyruvate is converted into lactate, or into lactic acid. This process of fermentation is going to take the NADH made during glycolysis and convert it back into NAD+. By converting the NADH back into NAD+, this is going to allow glycolysis to happen again. Each time glycolysis takes place, the cell is able to make 2 ATP. So overall, the process of fermentation has one goal. Its goal is to perpetuate the process of glycolysis and keep making 2 ATP at a time. Yeast cells undergo alcohol fermentation, which is exactly the same, except that instead of producing lactic acid, yeast produce alcohol and carbon dioxide. But again, the goal is the same, to regenerate NAD+, to perpetuate the process of glycolysis, to make glycolysis happen over and over again. So what happens if oxygen is present? If oxygen is present, the pyruvate will enter the mitochondria, and it goes directly into the mitochondrial matrix, where the next chemical reaction takes place. The next chemical reaction is called pyruvate oxidation. Pyruvate gets oxidized. It loses its electrons. The electrons go to NAD+ and form NADH. NADH, again, an electron carrier. It's going to shuttle the electrons, in this case, that came from pyruvate, to the electron transport chain. When pyruvate gets oxidized, some carbons are lost, and those lost carbons are going to be gotten rid of, are going to be lost as carbon dioxide. The end product of pyruvate oxidation is a 2-carbon molecule called acetyl-CoA. That acetyl-CoA, there's actually 2 of them made because you have 2 pyruvate, is then going to enter the Krebs cycle. The Krebs cycle also takes place in the matrix. During the Krebs cycle, glucose becomes completely oxidized. The last electrons that originally came from glucose, they are removed. And all the carbons that came from glucose, they're all going to be released as carbon dioxide. Since all these electrons are being removed, the electrons are going to go to NAD+ and form NADH. You're also going to have some electrons that go to FAD+, and FAD+ becomes FADH2. FADH2 is just another electron carrier, just like NADH. This process of the Krebs cycle is also going to produce 2 ATP, just like glycolysis. So if we look at these chemical reactions that take place, glycolysis right here, pyruvate oxidation right here, and the Krebs cycle right here. All 3 processes have the same purpose. Glucose is just further and further being oxidized. First, glucose is oxidized into pyruvate, then pyruvate is oxidized into acetyl-CoA, and then acetyl-CoA is oxidized into carbon dioxide. Each time electrons get removed, electrons are going to end up with NADH, an electron carrier. All the carbons that began with glucose are going to leave as carbon dioxide. Since glucose has 6 carbons, 6 carbon dioxides are going to be released as a waste product. So where do all these NADHs and FADH2s go? They're all going to transport the electrons or shuttle the electrons to the electron transport chain. The electron transport chain is the final destination of NADH. At the electron transport chain, a process called oxidative phosphorylation. Oxidative phosphorylation is how a bulk of the ATP is made during aerobic cell respiration. The electron transport chain is embedded within the inner membrane of the mitochondria. So if we're looking at this diagram here, this is the inner membrane. Down on this side would be the mitochondrial matrix, and on this side here is the intermembrane space. The NADHs are going to travel to the electron transport chain. The electron transport chain is made up of a series of proteins embedded in the inner membrane. At the electron transport chain, NADH is going to be oxidized. It's going to lose its electrons, and it's going to lose its protons, its hydrogen ions. The electrons are going to be pulled down the electron transport chain. They're being pulled down by oxygen. The only purpose of oxygen is to act as the final electron acceptor. All these electrons that began with glucose, they're all going to end up with oxygen, and that's because of oxygen's electronegativity. As the electrons are pulled down the electron transport chain from protein to protein, they lose energy. And the energy that's lost is going to fuel these proton pumps. These proton pumps, or these proteins, are going to pump all the protons, or all the hydrogen ions, that came from NADH into the intermembrane space. And they build up what we call a proton gradient, also called a proton motive force. This proton gradient builds up just like hydrogen building up behind a dam. Well, in every dam there's going to be a floodgate, and our floodgate here is a really important enzyme called ATP synthase. When all the protons build up, the gate is going to open on ATP synthase, and the protons are going to rush through the gate. When the protons rush through ATP synthase, this spins a turbine on the ATP synthase enzyme. And the spinning of the turbine is what's actually used to regenerate ATP. This process of the protons, the hydrogen ions, rushing from high concentration down to low concentration is called chemiosmosis. Chemiosmosis is directly what's producing the ATP during aerobic cell respiration, during the electron transport chain. When the protons rush through the ATP synthase, they're going to join up with the oxygen and the electrons, and water is going to be formed. This process overall, where the movement of electrons are used to regenerate ATP, is called oxidative phosphorylation. Oxidation, the removal of electrons; phosphorylation, the production of ATP. 32 ATP are made during this process of oxidative phosphorylation. A bulk of the ATP made during cell respiration is produced by this means. This concludes our video.